It says now I got
started.
the message, I think.
Let's see now.
Yep. Yeah, I see it now.
It's working now.
Okay.
Perfect.
Good. Perfect. And now I'll share my screen. Okay, so let's get started with this. Okay, so hello everyone. Thank you for joining us for another exciting fine seminar. My name is Adriana Maldonado. I am a professor at Universidad del Rosario in Colombia, and I am the host of the seminar this week. I'm really happy that you are all here today. And before I announce the speaker for today, I would like to make some announcements. First, I would like to thank our previous speaker, Tony DiFiore from University of Texas, for his great talk and discussion on agent-based simulation modeling for studying social evolution. And I also want to introduce our next speaker for next week, Michael Greiser. He'll be talking about cooperation and family living, and we'll be looking forward to his talk. Just a few reminders about the structure of FINE. We have a 50-minute presentation followed by around 1 hour of discussion. During the seminar, please make sure that your microphones are muted. The only time that they should be on is when you're engaged in the discussion. Then during the discussion, if you have questions, please type the question mark in the chat and I will call you on. If you're watching the stream on YouTube, please type in the question and someone will relay the question to me, or it will be asked at some point. We always encourage the students and early career researchers to ask questions, so please do it. And with no further ado, I'm pleased to introduce this week's speaker, Dr. Gloriana Chaverri. Gloriana got her PhD at Boston University in 2001. After that, she did 2 postdocs. The first one was in Western Michigan University in Michigan, and was done in 2006. The second one was in Boston University, Massachusetts, in 2008. And since 2011, she's been an associate professor at Universidad of Costa Rica in Costa Rica. She's currently based in the smallest campus of the university in Golfito, which is a beautiful small town as well. And then in 2018, Gloriana also became a research associate at the Smithsonian Tropical Research Institute. Gloriana's research focuses on understanding the environment's influence on social organizations, from understanding how groups are formed, how are they maintained, and the patterns of associations and interactions among group members. More broadly, she is interested in social behavior and sensory ecology, using neotropical bats as a study system. Currently, Gloriana has several projects that range from social behavior all the way to the interplay between climate change and physiological and sensory responses of insectivorous bats. For these projects, she works with collaborators from all around the world, and she also supervises a great number of students at all career stages, ranging from undergrad to doctoral students, and also postdoctoral researchers, especially from Latin America, but she has also had 1 PhD from Bulgaria. Talking about her publication record, it's very impressive and very diverse. She publishes in a wide range of journals, such as Current Biology, Animal Behavior, PLOS ONE, PROGB, Journal of Experimental Biology, Molecular Ecology, and also in some other applied journals, such as Conservation Science and Practice and Conservation Biology. And she's been really successful securing funding for her research since very early in her career. She's been funded by the National Geographic Society, IDEA World, American Society of Mammalogists, Bat Conservation International, Roost Small Grant for Nation Conservation, and the list continues. I think I'm just going to close it up here. And the title of the seminar today is Vocal Communication and Group Coordination in Disc-winged Bats. Gloriana, thank you for accepting our invitation, and the microphone is all yours.
Thank you, Adriana. And of course, thank you for the invitation. I'm really, really happy to be a part of this, wonderful community that, with people and present along with people that I have admired for so many years. So I hope I can also keep up with the expectation of everyone. And so hopefully you can see my presentation well enough. So I'll just get started. So like Adriana said, I'm going to present to you about this system of vocal communication that we found, specifically in a study system that we have been finding that it's very, very adequate to answer many of the questions that we've been interested in for many years. And before I start, I wanted to point out that, of course, like many of us, the majority of the studies that I'll be presenting are the result of a lot of collaborations and the effort and enthusiasm of a lot of students that have helped me throughout the years. So I want to acknowledge their role early on because I won't be showing the photos of them as I'm presenting. So I think one topic that has been quite fascinating to many of us in, I think, more recent years is the topic of group coordination. I think it's just becoming increasingly popular because we have many technologies that are allowing us to understand this process of how animals are able to coordinate where they move, how they move, where they forage or sleep. And so I think the study system that I've been focusing on a large part of my career since finishing my PhD, the disc-winged bat, is a great example of how animals are able to coordinate their groups. In particular, I'm interested in some of the behavioral mechanisms that are used to maintain this group cohesion. And the problem is that we still are in the early stages of to understand of our understanding of how animals manage this. How do they manage to stay coordinated or to maintain cohesive groups? And specifically, I'm interested in the topic of communication. What's the role of communication for maintaining group coordination or group cohesion? So I think what I want to show you primarily during my talk is how this system that I've been focusing in uses this system of communication for group coordination. But also I want to show you with a little bit more detail other aspects of this study system, and especially try to compare it to other species of bats that have been also studied in great detail, and show you how this species is so far rather unique in terms of what we know about their social system specifically. So first, I'll show you this study system, or the main focus of my talk will be on this study system, this species of bat that I show in this illustration, and this strong emphasis in my talk on the communication that it uses. And then I'll focus also a little bit on its roosting ecology, which I think it's really important to try to understand the process of group coordination. Then I'll also show you a little bit of the details of its social system. So I think that really helps us put in perspective some of the causes and consequences of group coordination. And then also I'll focus at the end of my talk on vocal personalities and what we have been gathering in terms of information on how different individuals produce more or are more vocal than other individuals. So first, I'll focus on the species in particular that I've been researching. So this is the disc-wing bat, and the species is Thyroptera tricolor. And it's a very small bat. It's about 4 grams, and they have also this charismatic, or this very interesting structure on their hands and on their feet, which are called the discs, or people also say that they're adhesive discs. And they're very important structures for allowing the bats to attach themselves to the inner surfaces of the leaves that they use for roosting. So it's a very, very important structure that explains a lot of why they use the roost that they normally use. They live on the lowlands of the neotropical region, and they feed primarily on jumping spiders and also leaf hoppers, but they also, their diet includes as well lepidopterans and dipterans. This species, it also has something interesting compared to other bats as well, and it's the process of in terms of the reproduction. Like many other bats, this species typically has only 1 pup per year, and females reach sexual maturity when they're 2 years of age. And the pups are born really small without any hair. They're very, very dependent on their mothers, and they're weaned at about 4 months of age, which is really old for a species of bat, especially one this small. Another very interesting thing about this species is that both male and female young tend to stay for a very long period of time, many, many years even, within their natal group. So they're highly philopatric, and so the groups are composed primarily of the descendants of the original mothers. So I wanted to show you one example of a group that we tracked for a few years, and this just shows the pedigrees that illustrate the matrilineal relationship among the group members. So like typically the females are shown in circles, and the males that are born, or the males within the group, are shown in the squares. So we can see what I did here is show different matrilines in different colors. So some of the original females that formed this group, which we named group 1, are shown in these 3 different colors. And what I'm showing is which individuals are being born each year. So for example, in 2007, this white female gave birth to 1 female, and whether this female stayed within the group or finally dispersed or not. But I wanted to point out, for example, in this case, to demonstrate this natal philopatry, or the pups staying within their natal group, for this female here, that this female only gave birth to males, and all of the males remain within their natal group for a very long period of time. And the other thing I wanted to point out, which is something that we're trying to study as well, is this interesting process that we haven't tracked for many years and for a lot of groups. But we've seen that there's a tendency that when individuals disperse, it's normally the mom with the pup of the year that we see that leave the group together. So I think this might be potentially a very interesting topic to look further into, to see how new groups are established, and if it's always based on the female dispersing with their young of the year. All right, so now on to the roosting ecology of this species. I think it's very interesting because from the perspective of a bat biologist, we always focus, when we want to understand social behavior, we always focus on what's happening at the roost. For bats, it's really difficult to track what they're doing far away when they're flying or foraging. We need technology that for many species is still not available. So when we want to understand social behavior and a lot of the patterns that I'll be talking about during my talk, we tend to focus on what's happening at the roost. And we know that bats use a large diversity of roost types, and they spend a large portion of their lives within these structures. So of course, there's a lot of interesting things that are going on when the bats are spending all this time with their conspecifics within these sites. So what's interesting about why the roosts are important and in terms of social behavior is that a lot of fitness related activities are conducted within these structures. So for example, we know, and in this also fine seminar, Jerry Carter presented a lot of the stuff that he's been doing with vampire bats. So we know that vampire bats, for example, share their food within their roosts. So this is a cooperative task that it's done within these structures. And we also know that for many species, or the majority of species that we know of, the mothers tend to their pups within their roost, and even they might be showing them how to forage, how to feed, how to handle difficult prey items within their roost. So a lot of learning processes may be taking place also in these structures. And then also we've seen, and we know that copulation happens very, very often at the roost. So I'll show you here below a video that shows the male that's on top and the female on the bottom. And the female has a 2-day-old pup. So, and the female is already copulating, it's in estrus. So they have a postpartum estrus. And this is another species that I studied for my PhD. So what you see is the male attempting to copulate with the female, and this is one of those instances when we think the copulation was successful. And they tend to attempt to copulate very often during the day and at their roost. So they're not primarily nocturnal creatures in some of the behaviors that they conduct, social behaviors. So what about the roosting ecology of the species that I've focused in, the disc-wing bats? So we know that they live inside these developing tubular leaves of plants such as heliconias, calatheas, and even banana and plantains. So they go inside the structure there. And when we see them, for example, if the leaf was transparent, we see that they're arranged in a linear fashion, and it's consistent. They place each other in a specific order, and it's a very common structure. You can find it very often in the field, and it's quite good at concealing the bats. So in this video, what I'm showing is how the bat would look inside. So there's a tiny bat inside the roost. And it's difficult to see. In this case, the leaf is a little bit open, so you can see it more easily, but normally they're found in more concealed structures. So in terms of how these roosts are dispersed within their habitat or the landscape, normally we see these sort of plant species found in patches. So we have this series of patches, and normally what we have is in each one of those patches, we see that they're occupied by a group of bats. And so we also see that, well, their roosting home range is rather small, only 0.19 hectares. And we've also been seeing that normally the groups occupy their own territory, and those territories, those roosting territories, are not joined to other groups. So they have this very well-defined area that's only occupied by a single group of individuals. And also something that's very important to keep in mind is that these roosts are very, very ephemeral. So they normally prefer a diameter of opening of around 7 centimeters. And the leaves can remain within this size for approximately 1 day. So this means that bats have to change roosts basically every day. Every day they need to find a new place to spend the next day. And if the bats do not select an adequate leaf during the night, and sometimes they will be found in leaves that are wide open, and they're extremely exposed in this way to potential predators, diurnal predators. So then it becomes really important that they have to switch to a new roost, even do it during the day when they're a lot more vulnerable to predation. So this is one of the most interesting things that I'll focus on a little bit later in my talk. So also, when we talk about this species social system, then I'll explain to you a little bit about this and also why this is relevant to my talk. So I show you that, within these patches of heliconias and calatheas, you normally see a single group. And the group size has a median of 5 individuals. So it's a fairly small group size, and it ranges between 1 and 11. Normally when it's 11, they tend to separate quite often. But yeah, the typical group size is 5, 6 individuals. And in a separate study, we also looked at how stable, how cohesive those groups are. So we tracked the same groups for a long period of time, and we generated the social networks to try to understand if there were, often exchanging individuals between groups. And what we see is definitely that groups or the same individuals within a group tend to stay together for very long periods of time. Even the same individuals may stay together 2-3 years with very few exchanges of adults between groups. So this is something also rather interesting about this species. And the other thing that, you probably already hinted that because the pups are staying with their mothers within their natal groups, that within group relatedness is very, very high. So we normally see relationships typical of, half siblings, but of course we see a lot of relationships, a mother and their several young. So this is also something quite interesting about this species. And the other thing is that, of course, they don't mate with their group members. They're all siblings, half siblings. And so normally what we find is that they go find mating partners quite far away, relatively far away, because we're talking about a species that has a very small roosting home range, but they normally travel 500 meters or more to find a mating partner. What I find quite interesting here is that even though they're not at all related to individuals that are found right next to their roosting patch, they go very far away to find a mating partner. So this is a question that has been fascinating me for several years, and why this is happening. Why don't they copulate with individuals that are closer to their roosting patch? Okay, so I'll start now making a few connections. In this case, this process of communication and how it relates to roosting, the roosting ecology of this species. So I told you that the roosts that they use for during the day, they normally open very, very quickly. And so when the bats are found that they, like even during the day, if they're flushed by a potential predator or they're scared, or even the leaf opens up during the day, they have to find a new roosting site. And this is something that happens quite often during the day as well. So when this happens, the bats have to fly away and try to locate a new roost site. And so obviously, maybe in this case, we will start wondering whether they use any sort of communication system to try to coordinate where they're gonna go next for roosting. And here I wanted to show you this very interesting study that I didn't really notice until after I had been doing a lot of the communication studies that I've done with this species. But it's a study published in 1974 by James Findlay and Don Wilson. And I really love these sort of papers when they describe in a lot of detail some of the natural history of a species and some of the observations that they did, which are very similar to what I started finding when I did my own study. And so they went out and captured groups of Thyroptera, and they released some bats, and they kept other bats within a cloth bag. And when they released the bats, they started flying very close to the cloth bag where they had other individuals. And this is exactly what I found in my initial studies. And then when they released, if one of the bats from the group found a leaf for roosting, and they released another set of individuals, they saw that immediately they went to the leaf that was already occupied by individual. So the exact same observations that we did, but now at my time, we had some of the technology to try to address this issue. What's going on. I had more equipment available to me to try to understand what's going on. So of course, this hints at the idea that there's communication going on, and probably acoustic communication. So to try to understand what's going on and how they're communicating the location of a roost site, we did an experiment in which we released the bats in the field, and we placed an individual within one of these tubular structures, and we wanted to see what sounds were being emitted. So we located microphones around the area where the bats would be flying, and we placed another microphone near the roost. So what we found is that the bats, when they're flying, in addition to echolocation calls, they're also emitting what we named inquiry calls. So they're very constantly emitted during flight. And then the bat within the roost, when they hear, when an inquiry call is emitted, then the bat within the roost may often produce these response calls, which are longer, more complex calls. And by emitting these response calls, the bats that are flying will very, very quickly enter the roost site. So I'll show you a video here. And what you're looking at a plastic, transparent, tubular structure that we often use for our experiments. And the camera is looking at the entrance of this roost. And you see a few bats flying around, and then one bat enters, and then the rest will do so. But what I wanted to point out that I also overlapped the calls or the signals that they used for this process. I lowered the frequency and stretched the time so you can hear them more properly. But the first call you'll hear, which is like pew, is the inquiry call, and then the response is like a really long call, and hopefully you'll hear that. But it's like the bat within the roost will start producing the response calls after it hears the inquiry call. It's also shown. And we also wanted to see if the bats were entering in a specific order, so we painted them with fluorescent powder, which is non-toxic. We knew that this communication system was very effective in driving individuals into the leaf, and we wanted to see if this communications was also really important for reducing this amount of time, now doing it in a more experimental, more controlled fashion. And specifically, we wanted to look at whether having a non-vocal bat, which is shown on the left, versus a vocal bat within this tubular structure was allowing bats or the entire group to locate the roost much faster. So for this, we did this experiment in a flight cage, and we released the rest of the group, and we want to see how quickly they would enter. So of course, like we expected, what we found is that when we had a non-vocal compared to a vocal bat, how quickly the entire group was entering, and when we had a vocal bat, the time needed was much shorter than when we had a non-vocal bat. So this is already very strong evidence that indeed this response calls are very important for maintaining group cohesion, and especially for finding a new roost very quickly, which is really important because then bats may avoid being conspicuous to predators much easily. Now, some of the majority of the studies that we've done have been based on understanding the response calls, but more recently we also are trying to understand the role of the inquiry calls, and for this topic, especially of group coordination. So what we normally see is that when we have two separate bats flying in the field and one of them emits an inquiry call during flight, then the bat that's flying far away will very, very quickly join this individual. So we wanted to see if, so the fact that bats are flying close to each other because they're emitting inquiry calls, they allow individuals to find each other during flight. Maybe this process of group coordination or flying close to individuals is allowing bats to find the roost much faster. So it's not only the response calls that are emitted from within the roost, but also being able to coordinate the groups during flight that maybe are allowing them to quickly locate a new roost site. So we first wanted to see if indeed the bats are coordinating adequately during flight, if they're able to stay cohesive during flight, and then what's the potential role of this coordination for locating and quickly and coordinatedly locating a new roost. And for this study, we did what we typically do for all of our research with Thyroptera, and this is something that always puzzles people because we always do our research during the day, because this process of coordinating to find a new roost site happens during the day. So we go out during the day and we locate bats with these extendable mirrors just to see if the bats are within and not having to disturb them. And if we find a group, then we'll bring it to the lab or a flight cage, depending on what we want to do. And so we, in this case, for this study, we brought bats to this white flight cage. And within the cage, we placed microphones and we placed a set of cameras on one side. And then we registered the flight of all the individuals within the cage. So with this, what we generated is a series of flight trajectories. And with the trajectories, we just wanted to see if the bats were flying cohesively, or if the mean distance between individuals was fairly short, or if the distance was larger between the individuals that are flying within the cage. And in this case, we also extracted individual trajectories from several groups. So some of the groups that we had the tracks for the individuals, we extracted these trajectories, and then we created these simulated flights. So we had these other simulated flights. This is a flight from 4 individuals that were taken randomly from other groups. And then we wanted to compare the distance between these real groups and the simulated or real trajectories versus the simulated trajectories to try to determine if flying close to each other was just, the result of social factors or other non-social factors if the distance between the real and simulated was very similar. So what we found is, in this graph, what I'm showing is the mean distances between individuals on the x-axis, and then the number of observations that we got, and depending on whether it was a real flight or a simulated flight, and we show the results for different group sizes. So here in this purple curve, we show that the mean distances between individuals is very short for the real flight types, and then for simulated, the distance between individuals tends to be much higher. So it seems like a logical conclusion that bats are being able to coordinate quite closely their flight patterns within, in this case, the flight cage, which we think extrapolates also what's going on in the field. And this is another video that just shows how coordinated I mean that the bats are. So it's a group of 5 individuals. They're not only flying close to each other, but also they change directions in almost at the same time. So this is something that we see very commonly also in the field. Okay, so we know that then the bats are flying cohesively in a coordinated manner, but then we wanted to see if this translates also to locating the roost site in a coordinated fashion as well. And for this, what we did is brought, again, the bats to this white flight cage, and we placed a leaf with some obstacles that you see as the strings. And we allowed the bats to fly, and then we recorded the time when the first bat entered, and compared that to the time with the final bat entering the leaf. So we wanted to see how these differences were compared to the type of the group that we did, and I'll show you this a little bit. So in this case, what we did is we created these real experiments where we released several bats with their original group size, and we measured the time difference between the last and the first bat that entered the roost. We also then created these artificial groups. And in this case, what we did is just mixed bats from different groups, and we created also different group sizes. And we wanted to see if there's some difference in how coordinated they are when they're familiar with each other, or they're non-familiar with each other in the artificial groups. And then we also released single individuals. So it's just one bat that had to find the roost within the cage. And then with these times that we generated for individual flights, we randomly created this what we call individual groups, which is just the time taken by each individual, and then did the same calculation of the difference between when the first one entered and the last one. And this comparison basically allowed us to determine if flying in familiar groups would allow bats to enter in a more coordinated fashion, or if just group coordination while entering a roost was possible simply by being with others in the roost. So, and this is some preliminary results, and this is again the time difference in seconds from the last to the first individual that entered. And what we see is that for both the real and artificial groups, we see that the time difference is much shorter. Then if we compare those random groups that we created based on individual flights, that the time difference is much, much bigger. So this just indicates to us that simply being with other bats within nearby is allowing them to locate a roost much faster, which provides some strong evidence for why just locating other individuals in the field using inquiry calls may allow bats to quickly enter a roost site. All right, so this other topic also relating the social system of Thyroptera with its communication. So we know that they have this system of inquiry and response calls, and that groups are very cohesive. So we needed to address the topic of whether these social calls have individual or group signatures, which are really important because they need to recognize each other if they're to maintain highly cohesive groups. And so if this is really happening, if there are group signatures, then individuals should be able to discriminate between familiar and unfamiliar calls to maintain these typically cohesive groups. What we found for both the inquiry and response calls, and I'm just showing here exemplars of inquiry calls, we see that, and this is 5 different calls from five different individuals. And there's definitely very strong individual signatures within both types of calls. We also wanted to determine if the signatures were acquired early or later during the process of ontogeny. So in the graphs, what I'm showing is the level of individuality of each one of the calls And basically it's just how likely we are to assign a call to a specific individual. So the top graph shows individuality for various age categories, which are measured in days, and then compare that to call individuality for adults. And this is for response calls and for inquiry calls on the bottom. And then what we see is that for all ages and call types, and both call types, individuality was significantly above the expected error. It just means that each bat has a very distinctive call. No, so we know that the calls are fairly different from each other. So we wanted to see also with experiments if the bats were able to discriminate between the calls emitted by familiar and unfamiliar individuals. So we did these experiments in a flight cage. Also, we want to see if bats in flight were able to distinguish from the inquiry calls from familiar and unfamiliar individuals. Then also if they were able to discriminate the response calls. So the bats that are flying, if they're able to discriminate between the response calls emitted by a familiar or unfamiliar individual. And then lastly, we wanted to see if the bats in the roost were able to discriminate the inquiry calls from familiar and unfamiliar individuals. When I say familiar and unfamiliar, it's just whether it's a group member or non-group member. And so I'm just showing here the setup for one of the tests that we're doing. And this setup, I'm with a couple of students and I have a bat in my hand and I'm about to release it. And we give the bats a choice to either go to either one of these setups on the sides of the flight cage. And so it's not very easy to see, but there's a box, a green box, and it's attached to the box is these plastic leaves that we use to simulate in the leaves. And within the box, we have one of the speakers. So we're broadcasting calls from a group and non-group member, and just we wanted to see where the bat was going. So in general, what we found is that the bats within flight, either when they're selecting to join individuals based on inquiry calls, or when they have to decide which leaf to go into, the bats seem to discriminate between these calls because they prefer to join the familiar calls or calls from group members. And then what was very surprising to us is that the bats within the roost did not seem to be able to discriminate. At least we didn't find a preference to respond to group members. So we started thinking whether the roost that the bats are using is somehow affecting the process of communication. So when we focused on this idea, whether they're able to discriminate when they're roosting and they hear an inquiry call, we wanted to understand if the shape of the roost was somehow affecting this process. So we have this structure, this tubular structure that very strongly resembles the acoustic horns that we humans use very often. And we know that acoustic horns not only amplify the sounds, but they also distort the sounds quite strongly. So to understand if this is happening in this communication system, we did an experiment where we placed a microphone within a tubular leaf or left a microphone outside a tubular leaf. And then we started broadcasting a series of inquiry calls from different frequencies from different individuals, and to determine if somehow the structure is affecting the signal or potentially affecting signal recognition. So what we found is, and I'm showing here the spectrogram and waveform for both signals, or just an exemplar of one of the signals that we recorded. And this is, one of the inquiry calls. And the important thing to notice here is the waveform on top. And it just shows how intense the signal is, whether the volume is really low or the volume is much higher. So I'll also present to you the recording of this particular call, and you can judge whether at least the sound seems stronger in this case for the control, or it sounds stronger for the experiment. So what we typically see was, what we typically saw was that there was a strong increase in the intensity of the call when we placed a leaf around the microphone. And so this is something interesting that we didn't expect to find. We were mostly focused on sound distortion, but we also saw that there's a huge increase in the amplitude of the signal that the bats are receiving within the leaf. And so I'm showing here the distance, for the microphone, but of course that simulates the bat within the leaf. So if we moved the microphone from the top all the way to the bottom, and when we recorded the calls, the inquiry calls, we saw that as the microphone was placed deeper within the leaf, the amplitude, the perceived amplitude was increasingly higher. And bats are normally found within the deeper parts of the leaf. So this is a good simulation of how they would be receiving that call if they were in their normal position. But then the most important thing that we wanted to focus on was this, whether we were finding differences in the perceived frequency in the experiment that we did with the microphone placed within the tubular leaf. So in this graph, I showed this difference in frequency. And this difference in frequency is, the recorded frequency versus the original frequency. If there's no difference in the recorded frequency with the leaf placed around the microphone, then all of the bars should be near the zero measure, or no difference in frequency. And I also present the original frequency of the call in different, in kilohertz for different calls, because we found this, we did this experiment for many different types of calls, all of which had different frequencies. And what we see is that for the majority of the calls that we broadcast or we use for our experiment, the perceived frequency was much different to the original frequency. So when the bats are emitting these inquiry calls and the calls are entering the roost, we see this increase in sound amplitudes, basically because the sound waves emitted by the flying bat get increasingly compressed as they enter the roost. And we also see this sound distortion because the leaf is acting potentially as a waveguide, and these waveguides create this confusion of mixture of different frequencies that have been bouncing off the internal walls of a structure, in this case, the tubular leaf. So we think that this sound distortion may be hindering signal recognition very strongly in this particular part of the communication process of Thyroptera. All right, so for the last part of my talk, I wanted to focus a little bit on a lot of the studies that we've been doing on vocal personalities. And this is a lot of my ongoing projects are based primarily on this topic. But since the FINE seminar public is a little bit more, well, it's still focused strongly on the topic of animal personalities, but also I wanted to focus a bit more on social system and other aspects of this species. Okay, so in terms of these differences in vocal behavior, until date, we've focused mostly on the response calling system, or this part of the communication system. So here, what I'm showing in this graph is different groups that we tracked for some time, and we had information about their vocal behavior, especially in terms of production of response calls. So what I'm showing on the y-axis is the number of calls that the animals produce in the series of experiments that we do. And I just want to show 3 examples of 3 different groups that tend to follow the typical pattern. Each one of the red dots represents one individual. So for example, in group 1, we find 3 bats that normally do not produce any response calls, at least from the several tries that we attempted with them. And a couple of other individuals will produce response calls in different rates. And we see the same for basically the majority of the groups. And this is something that's consistent, it's repeatable, and even for very long periods of time. So one of the topics that we wanted to address is, what's one of the potential causes of this variation in calling rates for the response calls. So we wanted to try to understand one of the potential costs of sound production. We focused on the energetic cost of this production of these sounds. So for this, we did respirometry experiments, and we placed a bat inside a chamber, and we measured oxygen consumption and this is just to show nicely how the setup was made, but the real setup is really messy, and there's a lot of cables coming in and out. So, but this is just to show that the bat is placed within one of these chambers. And then we measured oxygen consumption as a proxy to determine energetic cost. What we did in this series of experiments is we subjected bats to 2 treatments. The first treatment, and they were presented in different orders randomly. For one of the treatments, we just placed the bat inside the chamber. They were resting comfortably inside one structure that resembled a tubular leaf. And we broadcast no inquiry calls. The bats were just sitting calmly within the chamber. And this was to measure basal metabolic rate. And in the second treatment, we would broadcast inquiry calls, and we wanted to see with this, what's the cost of sound production. And we ran the trial for 10 minutes, each one of these treatments. And this is more or less what we expect, right? The basal metabolic rate, the oxygen consumption is a bit lower than for the treatment where if they were producing response calls. So we measured this increase in energy consumption from treatment 1 to treatment 2. And I'll show you what we found for this experiment. And here in the y-axis, I show increase in energy consumption. And we separated the results for males and females because we found some really interesting differences between them. And also I'm showing results for the non-vocal and the vocal bats, because even though we would broadcast the inquiry calls, some individuals were not responding, like I showed you before. It happens very often. And so for the non-vocal bats, what we see is that for both males and females, energy consumption is a bit higher, even though they're not producing any response calls compared to the basal metabolic rate. But then for the vocal bats, the increase in energy consumption is very significant. So we see that bats that are producing response calls are subject to very strong costs, and maybe not all individuals are able to afford these costs. So maybe this explains a little bit the differences that we've been finding in vocal personalities. We also wanted to see if these differences in vocal behavior during response calling were associated to how exploratory bats are. So we tested this by placing antennas in different roosts within the field. And we were trying to identify which group members, because they're all pit tagged, were entering the roost first most often. So the bat that enters the roost faster or initially would be probably the bat that's calling to the rest of the group and allowing the individuals to then find this roost very quickly. And what we see is there's an association between the number of calls emitted and the number of leaves that a single bat discovers when they're in the field. We also wanted to determine if differences in vocal behavior were associated to the use of personal or social information during the location of new roost sites. So what we did is release the bats in a flight cage. We know that these bats are either non-vocal or vocal from previous experiments, and we started broadcasting response calls from a leaf, and we wanted to see if there are differences in the time it takes the non-vocal or vocal bats to enter this leaf. And we did this in 2 trials for each individual. So for the first trial, we see when we compare the time the bat needs to enter the roost, we see that non-vocal bats are entering the roost much faster. and it takes much longer for the vocal bats to enter the roost. But then when we run the second trial, we see that there's basically no difference in how long it takes them. So what we think is going on, this is still in review, but we think that the vocal bats are less responsive to social information of the location of roost sites and more dependent on their personal information. So this is also an interesting behavior that we've been recording. So to finish up, I think my time is almost done anyway. I wanted to again focus a little bit on this study system and why we've been finding that it's rather interesting and potentially very unique among bats. There's not a lot of species of bats that have been studied in a lot of detail. I include some of the ones that we used for a review paper that we published a couple of years ago. So some of these species, we have information about relatedness and associations within their roost. And so in this paper, we were interested in trying to understand kinship association and social complexity in bats by using all of these examples. So I'll show you some of the most interesting differences between Thyroptera and these other species. And the first one is the association between relatedness and how often individuals are found with each other. And what we basically find is for Thyroptera here marked in red, there's a very strong association between relatedness and associations. So relatedness strongly predicts associations in these species, more so than what we find in other species of bats. Also, I show you that we did studies on group cohesion, and this review paper also addresses this and whether there are differences between Thyroptera and the other species. And what we find very, very commonly and very strongly differentiated from other species is that in Thyroptera, individuals rarely associate with other communities. So we think that from what I've been discussing and reading, comparing Thyroptera to other species, not only bats, but other mammals as well, we think that Thyroptera the social system is more closely related or similar to orcas. And we still don't know if potentially this use of a roost site, very unique roost site, is maybe explaining this difference. And I think to try to understand that, we can start focusing on other species that haven't been studied in a lot of detail, species of bats that also use this sort of roosting site. So with that, I'd like to thank you and thank you for your attention.
Thank you, Gloriana. Thank you very much for such a great talk. I think it's time for questions, and I just want to remind people that if you have a question, just type in your question mark in the chat. YouTube is about 1 minute delayed, so just make sure to get your questions on the YouTube, and we'll make sure to have them here in the Zoom. We'll get started with Tomislav. Tomislav, please. Don't forget to your introduction.
This amazing talk.
Thank you.
I was very interesting about your study system, because when I was doing my PhD study, I happened to stumble upon I was doing my PhD on sperm competition in salmon and trout. Mm-hmm. And then I found that Chiroptera have very interesting sperm. Mm-hmm. So my question is about sperm biology. Ooh. Which I assume is not your is not your field of expertise. Mm-hmm. But however, I would like to pose a question, because in that time I found a paper from 70s, which I cannot remember now the author, but I learned that Thyroptera sperm is quite long-lived, meaning that it hibernates, that it can survive the winter period. Now, after your talk, I realized that your study system is matriarchal, that is females that decide, that is a very small fitness output. There is only a single offspring. And my question then is, do you know anything about sperm competition in your study system? Because what I find interesting is that they are, even though they are close, they are not related. Which means that it should be very much field to be explored just within this relativeness between individuals which are relatively close and still not. So do you know, and can you tell us anything about sperm competition in the bats you were studying? Thank you.
Sure. I'm not sure if there's even, I'm trying to remember some studies that have been done on sperm competition in bats. And I think there's, I don't know, I can't remember of any papers that I've read about that topic. So it's still a very elusive topic for bats in general. I unfortunately, for my study system, I don't know anything about, even sperm morphology, sperm competition. But I'll, just so I don't leave the question like just with this very simple answer.
Thank you. I appreciate it, but it was just suggestion. I mean, because it seems that there is a quite a span of work possible to be done.
Yep.
Because bat sperm are extremely long-lived, and they hibernate, and given the social system, which is matriarchal, and there is quite a possibility to investigate this, because you can pose hypotheses whatsoever, I mean, about relativeness, about why do they not copulate with the close neighbors, despite though they are not related. I mean, so that was just a suggestion, but thank you for your answer. Yeah.
No, but I wanted to expand a little bit more on that. Just, I normally don't, of course, we all normally don't have time to present all of our findings or even, some of our observations. But the work that Findlay and Wilson did in 74 and even in later papers, what they found something interesting that we haven't been able to address. And it's the fact that females seem to be in estrus around August, September, for example. And then normally gestation happens fairly quickly in many species of bats, unless they have some sort of reproductive delay. And what we find is that even though they're in estrus in August, September, we start seeing the pup develop, or at least we can palpate the abdomen, and we notice that the female is pregnant, but we notice that in January, February. So there's probably some reproductive delay also going on in Thyroptera in this study system, but we haven't, haven't addressed that. So I think there's a lot of potential for.
But do you know there is sperm competition within the system, or I mean, do you know
bats
that?
in general, definitely. So there's also this interesting book chapter that some colleagues did, that they measured. So they compared the mating system with the size of the testes for bats, and they found, of course, as predicted, that you know, this.
Yeah, but I mean, but the question is, how many direct observations have been done, you know, and that is a. But that is the question. I mean, I hope somebody will get money to investigate.
Yes. That's, yeah, definitely. And we also recently started looking at some of the genetics. We have very little funding normally for our research, and now we have some money for addressing relatedness between some of the, and looking at when the pups are born. For example, are the females mating with the same males constantly year after year? We don't know that, and now we have the money to try to address that question. So it's definitely very interesting.
Good luck, Dad.
Thank you.
Thank you.
Sure.
Great question. Thank you, Tomislav. Just as a reminder, we're trying to build community, and so please state your name and your affiliation so we get to know each other a little bit more. Our next question comes from Lindelani.
Hi, everyone. My name is Rindelani Makoya, and I'm a PhD student at the University of the Witwatersrand in South Africa, working on the costs and benefits of solitary living in the Bush Kururet. Thank you so much, Gloriana, for the talk today. It was very interesting. I was very drawn to the topic about the what do you call it, the leaf that the bats use for the roosting. And I wanted to know, what are the threats that are faced by these bats in terms of habitat destruction and changing habitats or changing environments, and how do the bats deal with that, and what is their threatened, what do you call it, IUCN listing in terms of that? Are they threatened, or how do you,
I think a large portion of bats are not considered threatened. In Costa Rica, I think there's only 1 or 2 that are, and Thyroptera is not. What's interesting is that Thyroptera is really a very common species in the majority of areas, but because we normally don't capture it in mist nets, they're very, very good at avoiding the mist nets. Then people used to think that it was a very, very rare bat, but I think it's not that. It's fairly common. But it's a very interesting question, because even though the plants that they use for roosting are common in many areas, these areas are normally the product of trees falling down, or these plants just dispersing naturally around rivers, and so the areas, or these patches, tend to suffer very commonly from normal succession of the forest, and the plants disappear, not very quickly, but then eventually they'll stop producing enough leaves to continue being a suitable habitat for bats. So I haven't really looked at what the bats do eventually, or these groups that tend to remain very faithful to these patches. We don't know what happens in the long term, but I know that one of the previous colleague, Martin Vonhof, he noticed that for his field site throughout time, the plants were disappearing, and so were the bats. So the bats were left the area when the leaves were very scarce. We also did an experiment, and we wanted to see what happened if the plants were cut. The habitat was cut for a few groups, and what we notice is that normally the bats are they have these cohesive groups. They stay with each other day after day, very, very often. And when we cut the plants that they use for roosting in a patch, they started to separate more commonly, which also provide some evidence that this process of communication may be really important for them to remain cohesive, because when we cut their habitat and they have to fly really far away to locate new leaves, then they start splitting up. So this is more or less what we know about this topic of habitat disruption.
Okay. Thank you so much.
Sure, thank you.
Thank you, Lindelani. Just to make sure, we do have a question on the YouTube channel, but it's here on the list, so don't worry, we'll get to it in a moment. So next question comes from Clara.
Hi, I'm Clara B. Jones, retired social evolution. This is beautiful work. Your methodologies, plural, just make me want to remind the graduate students that the last 3 talks that we've seen show the combination of descriptive work, natural history work, and then using simulations, not only to test your data, your results, but also to conduct experiments. And this would be, a great advance to the field if more natural history students studies would include this, in addition to doing invasive studies, experiments in the field. And just one more comment to graduate students. If you go back in the mammal literature, you will see that field experimentation is very, very common in mammalogy. Now, my question, especially translocation experiments, by the way, my question is a very brief one. You begin your talk by showing a video of birds swarming, always a very impressive phenomenon. Can you say a little bit about how what your results tell us about the capacity for swarming?
I think it's widespread, definitely. And I think, it's done in situations where the species is very prone to predation. So I think in general, the primary pressure to create this swarming or these highly coordinated flight patterns, I think might be predominantly predation. But I still don't know. Go ahead.
Is your point the connection, or to connect the communication factors to communication's importance in swarming generally?
At least, for example, based on what we found in our system, we don't have a lot of evidence that communication within very short ranges, at least acoustic communication, is very relevant. So I think the second-to-second movements that must be achieved to be able to follow each other need to be from a sensory modality that provides information much more quickly. And so we think that it's possible that the bats, because they're doing these coordinated flights during the day, that they're actually able to accomplish them based on vision. So even though we think vision is not that important for bats, I think it is in this case. The other option is that they're following the echolocation calls from group members, which can only be heard in very short ranges, because they're emitting them very, very constantly, that perhaps echolocation has something to do, but I think for these very close and agile maneuvers that they really need, you know, a sensory modality that's much faster. And I think that's vision. So
Great answer. Thank you.
Sure. Thank you, Clara. And I agree completely also with the need to incorporate observations that we do in the field with our experiments and to generate really a robust, I don't know, knowledge base or information about a study system. We need to include and incorporate all these different facets of research. Go to the field, look at what your species is doing, run experiments if you have a question that you cannot answer by just looking at what they're doing in the field, and also use modeling because it's a very strong way to understand also some of the results you get from experiments. So good advice.
Just let me say one quick thing. This, of course, relates to the work that Ian Cousin is doing with his group, using especially math modeling, but also field observations. So he should really know about your work.
Yes. Thank you, Clara.
Thanks a lot.
Thank you, Clara. Our next question comes from Teresa.
Hi, so I'm Teresa Schabacker. I'm doing my PhD at the Natural History Museum in Berlin and the Free University in Berlin. And I'm currently in Costa Rica and Santa Rosa conducting field work on Glossophaga Handley, as we now call them. And thank you very much for your talk, Gloriana. Super interesting to me.
Thank you.
And I actually have two questions. So you mentioned that there was a relationship between the number of response calls and the exploratory behavior, right? It was just a bit too quick for me. Was it a positive or a negative relationship?
Positive. Yep.
Sorry?
A positive relationship.
One. Okay.
Yeah.
So the more exploratory bats were also the ones that were more response calling, right?
Exactly.
Okay. Because to me that makes lots of sense, because it would make sense that the bats that enter such a roost first are the ones that are calling, right? Response calling more to the group. That would be beneficial for the whole group, right?
Yeah, exactly.
So the other question that I have is like, how is that non-vocal behavior maintained? Do you think, is it maybe because of the high relatedness in the groups, or what is do you have a theory about that?
Yeah, and that's a very good question. We've also been trying to address that issue. Why are bats, staying quiet, even though it's really important for them to use these calls to maintain cohesion? And I think one of the experiments we want to conduct is whether having a lot of vocal individuals, maybe spread out, maybe that could confuse the process of group cohesion. So I think because only one or two individuals are really needed for this group coordination in terms of where they're going to roost, then we think that having more it doesn't really increase the chances of them being able to locate a new roost site. This is a very complicated topic, because then some of the individuals are, investing a lot of energy in trying to help coordinate the other group members. So this is also a topic that we want to address a bit more. Why certain individuals are doing that? We addressed a little bit of that with the energetic costs, but we also want to explore a bit further and try to understand why that's happening. Yeah.
Great, thank you so much. Because it's very interesting to me, because the Glossophaga species that I'm working with, I'm looking at individual differences in exploration behavior and risk-taking behavior, and I see the same thing. So I see that there are bats that are vocalizing in my trials consistently. Like, this is still preliminary data, I'm still in the data collection phase, but there are some individuals that consistently produce social calls and some that never do that. And the calls that they are mainly producing we term them alert calls. So it has kind of a relationship with like vigiliant behavior in this, and I'm just wondering about this. So yeah, very interesting to see your work there.
Thank you, Teresa.
Thank you, Teresa. Our next question comes from YouTube. Karsten.
Yes, hello, I'm Carsten Schradin, at the senior in Strasbourg, France, studying social evolution in mammals comparatively and in small mammals in South Africa, doing field work. Thanks for a great talk, Gloriana. I first have a question from YouTube, and then I take the liberty to ask a question myself. On YouTube, Brandy Cristiano says, "Hi, Gloriana. I'm Brandy, a PhD student who works with Rachel Page. Are the inquiry calls of these bats similar to the calls that Artibeus jamaicensis make when they get caught in nets and HE bomb you?" I don't know what is HE bomb. You probably know because you're a bad person.
Ooh, I didn't get that last part.
HE bomb. I don't know what is HE when they HE bomb you. I don't know what it means.
Okay.
Okay, I thought maybe it's something that all bad people know.
Maybe Teresa. Teresa raise her hand.
What maybe they are referring to is something that I have experienced, that when you have an AJ in your mistnet and they start producing the distress calls,
okay,
there are just lots and lots and lots of other AJs flying in your net. That's what I would be suspecting.
Okay, thank you. Now I pieced it together. No, they're not similar. So these inquiry calls are very short. And they're like frequency modulated, and they're not very noisy. They don't have a lot of entropy. But then the calls that are emitted by bats that are tangled in a net, or when you're handling them, they produce another type of call that's normally referred to as distress call. So the distress calls are, very, very loud, we can hear them. They're within our hearing range. And they're also very noisy. They have a lot of entropy, so they're not similar. And I think that's another topic that we wanted to address, to see what's the adaptive value of different forms of calls that we're getting for different species, in terms of social calls that the bats need to transmit in very long distances. And we think that the inquiry calls have a very interesting combination of acoustic parameters that allow them to travel really far away. They're a little bit on the lower side of the ultrasound range. They're around 20, 30 kilohertz. And they're not very complex calls. So we think that they are able to travel very far. And although the distress calls are very, very loud as well, but they're not. They're very different.
Okay, thank you. Then I have my own question. It's with regards to this phenomenon of cohesiveness, of social attraction. Obviously, living in groups must be beneficial for them, but it's not very special. I think most bat species somehow roost, probably because of benefits of predator avoidance or something like this. But in that species, it seems special that they're really so attracted to each other individually that they even fly together, that they want to be together with these individuals. Do you think this is because of their special ecology of roosting in these leaves, where only a few individuals have space, and this forces them to form groups, and then they prefer to form kin groups, and because they cannot, like bats that roost in large colonies, choose anyone, this is the driving force of this strong social attraction to specific individuals, or what is the explanation?
Yeah, this is at the heart of many of the studies that we've been trying to do, try to understand why they're so cohesive. There's other species that we know use a similar roost, and as far as I know, we don't have a lot of information on whether they're very cohesive or not. There's another in Southeast Asia. There's Caribula hardwicki, another species that uses this tubular structure, the one that I showed at the end that has the butt sticking out. They roost on the other way compared to Thyroptera, and they're solitary. So of course, there may be a lot of benefits to roosting with each other. Like you said, the typical, I don't know, let's say thermoregulation, even though a lot of people don't believe that this is something important in the tropics. I kind of do, but yeah, this is an argument to be made. Avoiding or being able to detect predators more easily. But the fact that they really prefer to roost with the same set of individuals, I think it's another topic that may be not directly related to the type of roost that they use. But I think it's an ideal combination of needing a resource that's scattered throughout the landscape. It's not always very easy to find. Maybe sometimes it's scarce. And then having the help of other individuals, and in this case, highly related individuals, may be just an easier way to be able to locate these resources. So this is, again, something, and something that I forgot to tell the community is that I welcome any possibilities, any research ideas, especially considering that many of you really strongly focus on the topic of social systems, and to try to help us understand why this is happening, why Thyroptera has such cohesive groups and highly related groups. Whether this is something that has to do with the roost, we still don't know. But definitely one of the main things that I'm interested in is trying to understand if there's a relationship between these two. And I'm not sure if I answered your question, Karsten.
No, you answered it. Yeah, we need more, maybe also comparative approaches to be able to understand this. The solitary species you mentioned is quite interesting. Of course, it would be the other way, the other tactic how to deal with this by being more dispersed. As nobody else has a question in the chat, I take the liberty to ask another question. Maybe you mentioned it when you were talking about these roost ranges. Maybe you mentioned it. But does this mean these are ranges determined by where they're roosting, but the foraging ranges are actually larger, or?
Yep, definitely.
So that means that there's no overlap in the roost range, but in the foraging ranges, there might be overlap of neighboring groups, but it's more difficult to get data on this because to put loggers on these small animals is difficult.
Exactly. Yeah. From what we know so far, the foraging ranges are much larger, and there may be also overlap. We don't know. And like you mentioned, the difficulty of tracking these very small bats is something that, the progress that's been seen in other bat species, we still haven't seen it because of the size. But that's it. Exactly.
Okay. Thank you.
Thank you,
Thank
Karsten.
you, Karsten. We've got another question from Tommy Slav. And
Yes,
I encourage students.
Thank you. I just wanted to adjoin to the previous question about relativeness between members in the group. My question is your species threatened?
Not really. At least from what we know, it's a fairly common species, and it's not,
okay.
At least from IUCN, it's not considered threatened.
Why do I ask that? It's because if they were threatened, and if they have managed to survive through bottleneck, then they could have purged deleterious alleles. So I mean, in that case, it could be that they have survived the bottleneck. So if they are not threatened, then it is even more interesting from a population genetic perspective, because then they should have or hold within populations deleterious alleles that are not purged. So I mean, that was just a thought.
Okay.
About the reasons. I mean, why should they still be so closely related and still be so close in a space?
And that would also explain because they mate. I don't know. Well, not necessarily.
But there is, that is what makes it interesting for me, because I mean that then the sperm competition comes in, you know. I mean, do they have sperm competition? How often? If they produce only one pup per year, I mean, how often there is extra par copulation in that case? Or are they monogamous? Are they polygamous? Do you know anything about that?
No, this is one of the questions we want to address now that we have a little bit of money for genetic analysis. So we've been tracking the pups that are being born year after year from the same females, and we want to see if those pups, you know, have the same father.
Because, I mean, from my point of view, it will be extremely interesting, because, you know, I happen to know that they have hibernating sperm and they can survive. Now I will not claim it 100%, but 75% a year, you know, after the winter. So it would be really cool to see. I mean, whose sperm really are fathering the single pup.
Yeah, that's true.
But thank you.
Sure, you're welcome.
Thank you, Tomislav. Well, I'm gonna take the advantage and ask a question myself. And meanwhile, I'll let some time for others to think about more questions. I have two questions indeed. The first one is a follow-up on Theresa's question. And I was wondering if these non-vocal phenotypes, can they actually vocalize? So they decide not to do it, or is it something that is fixed?
So we know, and this is something also that I didn't mention, that we think that the response calls are like come from isolation calls that the pups emit when they're much younger. So what we have noticed is that all of the pups, when we produce or we broadcast inquiry calls, all of the pups produce something that's very, very similar to a response call. So, and this is done by all the pups. But then when they start aging, then we get these distinctions. Some of them remain very vocal and the others just go quiet. And we also, in support of that, we also see that males differ in how vocal they are, depending on whether they're in reproductive season or not. So we know that, for example, males, when we sample males and we do these experiments and we broadcast the inquiry calls, and the males are non-active, they are not in spermatogenesis, and we broadcast the inquiry calls, and let's say half of them respond and half of them don't. But then we do this also during the reproductive season, and then we see that all of the males are responding. So we think that all individuals still retain the capacity, or at least they can associate hearing an inquiry call and the need to produce then a response call. So, but for females, we're not very sure about what may be happening. But at least for males, we know that they really do respond, especially if they're reproductively active.
Interesting. And the second question, it's related to the social system. So you mentioned it, and you also showed some social networks associated there, but you actually didn't went very deep into it. So I was wondering is there any, like, anything in particular about the social structure? Do they have like preferred partners? Do they roost together? Like the order resembles the strength of the social bond? Do they call each other like more often if they are more associated? I don't know. Like,
it's very interesting because because this is exactly what we're doing in a very large project that we have right now. So for example, this is something that I hadn't thought about, whether the strength of the association is somehow also related to how close they're roosting with each other. So we know that they, like I said, they position themselves in a very predictable manner. And so normally we would see the same individuals right next to each other, but we have no idea whether this also predicts, association in the long term, and association in terms of sharing the same roost at the same time. And then the other is also the study that we're doing right now, for which we have some money to do the genetics. We're looking at whether they prefer to respond to individuals that are more closely related, or to individuals that they're more familiar with. So they normally share the roost with an individual, probably because they're calling to each other a bit more. So this is something that so far I cannot answer, but we hope to have these answers in a couple of years. So maybe I'll present it again soon.
We'll love to hear that. Okay, so we have a question from Suleima.
I can't hear you, Suleima.
Sorry about that. I forgot to unmute. Suleima Tang Martinez, retired from the University of Missouri, St. Louis. And I have a couple of questions. One is whether there's any evidence of chemical communication, scent marking. I'm wondering if there is, whether there could be some recognition going on, especially in terms of how they space themselves inside the leaves, having to do with chemical, biological odors, essentially. So that's one question. And the other question you may have answered already, and I may have missed it because I had to run off and take care of the dogs. What is the situation as far as predation? I mean, obviously, they're roosting in these very interesting structures for a reason, and I'm just wondering if what predators they have, and if you think this could be an adaptation to predation.
So I'll answer this last one first, if that's okay.
Sure.
Bats, we know in general that they're not strongly affected by predation, especially when they're foraging at night. They can fly and evade predators very easily compared to many other groups of mammals. In the roost, we certainly know that it's a possibility. We often see capuchin monkeys and birds of prey that may be potential predators, but we think that within the roost, it's very difficult for these potential predators to find them. And we always know that it's in the process of roost switching, bats being flushed from their current roost, or just the roost is opening up very quickly, and they have to find a new roost site. This is when predation might be happening, and they're very clumsy flying, and when you see them during the day. So definitely there's a possibility of, especially diurnal birds of prey, but also monkeys have been seen to prey not on this specific species, but others that use similar, plant-based roost structures. And what we think is that in terms of roost selection, what we normally see is that the bats are selecting the roosts that are the tubular leaves that are very closed. So there's a trade-off here between being able to access the entrance of the roost, which is fairly small. The bat barely fits on the entrance. So there may be some accidents when they're flying and approaching the leaf, but they seem to prefer these more closed roosts, possibly because when they go inside the roost, they're very, very difficult to see. And this difference between the leaf being wider or narrower makes a huge difference in terms of whether you're able to see them, for example, in our case with the extendable mirrors, if we're able to see them or not. So I think that in terms of roost selection, that's the main way that they potentially avoid being detected by predators.
And so the leaves are formed in that shape. The bats are not doing anything to...
Exactly. Mm-hmm.
Okay, because I know, in some insects, they actually manipulate leaves and roll them up so that they can use them as a shelter.
Yeah,
So,
exactly.
So what about the chemical communication question?
Yep. So for bats, I think in general, we we're still developing this. I think there's a lot of new research that's being done on chemical communication, but unfortunately, there's very few. In this case for Thyroptera, we really don't have information about whether they're able to communicate through olfaction, or if this plays a role in, like in your example, of whether they place each other in a specific way based on chemical communication. But I think it's definitely a possibility. I'm not great at trying to understand this topic of communication, because I think we require certain other techniques, although experiments can be done as well, to understand olfaction and chemical communication. But that's definitely a possibility, but we don't know yet.
Yeah, but as far as you know, or as far as it's known, they have no specialized scent glands, or I know sometimes in bats I've seen them called crusts. They don't have anything like that in the species.
No, nothing obvious.
Okay.
Yeah, especially when you compare them to the glands, exactly, that they're used to create the crust in, the frog-eating bats, or,
Right.
There's so many species that have these interesting glands, but Thyroptera, there's nothing obvious.
Yeah. Okay, thanks. And
Sure.
Fascinating talk.
Thank you, Zuleima.
Yeah.
Thank you, Suleima. Well, I don't see any other questions in the chat. And I think it's been a fascinating discussion. So I'm just going to go ahead and stop the YouTube